You have seen that allele frequencies do not change in randomly mating bisexual populations, where each individual carries both male and female reproductive organs. Unisexual species, on the other hand, have separate male and female individuals. It is slightly more complicated to show the dynamics of allele frequencies in unisexual organisms compared to bisexual species. This is so because when we calculate the genotype frequencies in the second generation, we will have to take into consideration all 9 possible matings, that is, all 9 possible matings between each of the 3 parental genotypes in each of the sexes. We see them in the upper row and left column. Then each of the matings must be weighted by expected genotype contributions of each such mating. I will show you how. In this table, we see the fraction of total offspring contributed by each type of mating. We can ask, what will be the frequency of the big A dominant homozygotes in the next generation? To calculate this, we need to consider the frequency of each type of mating multiplied by expected genotype contribution. For instance, the probability of a big A homozygous male mating with a big A homozygous female is p squared multiplied by p squared, that is, p raised to the power of 4, as seen in the upper left corner of the table. All of the offspring will be dominant homozygotes. Next, homozygous offspring of the big A type will also be produced in matings between big A homozygous males and heterozygous females. The probability of such matings occurring is 2 times p raised to the power of 3 times q. However, only half of these matings will result in homozygous big A offspring, so the total probability becomes p raised to the power of 3 times q. If the father is big A and the mother is little a, then no big A offspring will be produced. Now we can evaluate each mating type in a similar manner. When the mother is big A homozygous and the father is heterozygous, then dominant homozygous offspring will be produced with a probability p raised to the power of 3 times q. If both parents are heterozygous, then 1/4 of the offspring will be dominant homozygotes, and the probability of this occurring is p squared times q squared. If one or both of the parents carry a recessive little a genotype, then none of the offspring will be of the big A type. All of this can be summarized as follows. The frequency of the dominant homozygous genotype, big A, in the next generation is equal to the sum of these weighted mating probabilities. And again, it is fairly easy to show that this equals p squared. The genotype frequencies of the heterozygote and the recessive homozygous genotype can be calculated in a similar way. The genotype frequencies have not changed. We still have Hardy-Weinberg frequencies. So this is the most important lesson from the Hardy-Weinberg model. Random mating does not change allele frequencies. Therefore, Hardy-Weinberg equilibrium implies stable allelic frequencies, the opposite of evolutionary change. In the next lecture, you will learn more about implications of the Hardy-Weinberg model.